Right, ladies and gentlemen, thank you very much for coming. It's a pleasure to introduce myself and to also introduce a little bit what we are doing. No, everybody's here. And also to introduce a little bit what we are doing in my group. So my name is Jan Roelof van der Meer. I'm Dutch, but I work in Switzerland already for quite a while. Right now I'm a professor at the Department of Fundamental Microbiology. Don't worry about the name. We're not so fundamental. That's how it is called now. I'm also head of the department currently, and our department has 7 professors in microbiology. So this is the building where we're at the university. It's called the Bio 4. So they hired some people to make fancy name instead of building CC 123 or something. So we have fancy names. It's a green building, and it's very close to Lake Geneva, which is a really wonderful place. And you have the Swiss and the French mountains in the back, so it's really a pleasure to work there, I think. So the current research topics in my group, so overall we're working on environmental microbiology. We are basically interested in 3 topics. One is called the biology of integrative and conjugative elements. I can tell another time about this if you're interested. It's a bit a biological topic, not very engineering or so. And then part of what my group is doing is we're working on what's called bioreporters. So we engineer bacteria to sense their environment for specific target chemicals and then produce signals that we can easily measure. And some of those are used in assays that we believe are can be very useful. And then the third part of what we're doing has to do with microbial ecology and bioremediation, a variety of aspects. Some of which I would like to present to you today. So basically, we are convinced that polluted environments, it's a bit, in a way, it's a tragedy, you know, polluted environments, we're stuck with all this dirt and junk and stuff in the environment. But from a biological perspective, this is a fascinating environment because it puts a lot of selective pressure on the organisms which are there. Obviously also larger organisms, but these can often not adapt within reasonably short times. So then you get dying fish or algae or whatever that are dying. But microbes can adapt. So, and basically there's 2 aspects that are interesting here. And one of them is that polluted environments are often toxic. So you get selection for organisms that are resistant to certain compounds, which has some similarity to antibiotic resistances, which is nowadays such a very big thing in all kinds of models, and we're pretty concerned about this, but we should be concerned about pollution in general as well. And the second aspect is that the pollution can serve as a specific energy substrate or as a carbon substrate for microorganisms which are living on that site. And that can even go so far that you can select from mutants, all kinds of mutants, spontaneous mutations inside an organism, but also things that where genes have transferred from one species to another, so that you get new strains that can then actually profit from this pollution and proliferate at expense of the pollution. So there's a lot of things that you can do with this. Some of the questions that we like to address is, how comes that certain bacteria are so stupid to eat, I don't know, toluene or chlorinated herbicides instead of sugar or amino acids? So what makes them so special that they can actually do this? And then the second question, so can we find evidence that bacteria actually evolve to pollution so that they accumulate changes which help them to withstand the pollution, to expel the pollution or to degrade the pollution? And if this is the case, then what kind of mechanisms do we find? And we have looked at a number of systems, including herbicides, pesticides, and there's some pretty interesting stuff where you get genes, mostly genes that are assembled within one organism that enable this organism then to use such a pollutant as a sole unique carbon and energy source. Then a third question that we like to address is, so if we find organisms that degrade something useful, is that actually going to help us? So can we apply these organisms again by, for example, putting them back into a site? Or can we just leave the things as they are and then hope that nature takes care by itself? And if we have this idea, how should this actually be done? And that's a little bit what I will be talking about today. And then finally, we're also interested to see what are effects of pollution on microbial ecosystems themselves, because, as I said, the selective pressure of the toxicity is very, very important, and that means that sensitive organisms will disappear. This will lead to changes in ecosystems with certain effects that often are not very well understood. So the topic I would like to tell you about today is this idea that perhaps it is possible that if you have bacteria that you think can use a pollutant and completely metabolize it to something completely inoffensive, like CO2 and water and chloride or something, is it possible to use this on a site? And this is work that we started in a large framework program a few years ago, and actually continues still now, because there's a lot of things that we want to do. And the idea is basically similar to what people are proposing for probiotics. So you can just dump your organisms on here, and oh wonder, you get back to fully restored nature, right? It would be nice if this is the case. Often it's just a matter of time scales. Obviously, we want that the pollution is gone yesterday, and we don't want to wait 50 years for it to slowly recover. And the idea with introducing these organisms is perhaps that you can speed up such a process so that you don't have to wait this long time. Actually, I think that many of the questions that we are asking are similar to the idea of probiotics. You know, you have your complex gut system, so perhaps in some cases this is disturbed, so can you drink something and then restore it? Or is this just, I don't know, completely outrageous idea, everybody proposed it, but it cannot work for this or that reason? We just don't know, I think. So in a way, it's approaching similar questions. So what I would like to discuss with you today are a few experiments that we tried to set out, first in this European consortium and then continuing in the lab, just to try to tackle that problem from a more, reproducible technical aspect. So the idea that we have, and the idea that's behind all these kind of technologies, is that you have a bacterium of interest. This may be, I don't know, maybe a lactobacillus in the case that you're talking about your gut, and maybe a pseudomonas and you're talking about the environment. You grow it in the lab, perhaps you have isolated it from a site somewhere, and then you want to introduce it back. So what's it going to do? If this is a soil, this can be pretty complex, because soil is an extremely complex environment made out of not only physical, chemical, very complex, but also lots of other bacteria that may be there and so on. So for the aspect of the process, there's a lot of practical questions that are important. You know, is this strain actually surviving if you put it in, or does it die straight away? Is it actually degrading something? Can it actually recognize that there is a pollutant which you want it to degrade? So lots of practical things that we have no real clue about. And then obviously there's also, I think, a number of more fundamental questions. For example, do bacteria realize that they're in a different environment? We like to grow them in the lab, in flasks, or perhaps in microfluidics, and God knows what kind of system you want to have, a 9-6 well plate or so. But do the bacteria actually realize that they're there, as opposed to being in an environment like a soil or a sand or on your skin or on a plant leaf? And if they realize, so what are the things that they see differently in those environments? This may be very important, because it tells us, okay, well, perhaps it's not a good idea to grow it in a chemostat there, because this is not really what they need to do when they are in a sand. Then there's a number of general things like, okay, if we want bacteria to survive, is there something general about certain strains that help them to survive and that enable other strains not to survive? Other strains may be inherently less capable of surviving when you put them back into the environment than others. The typical example is, I don't know, is anybody of you taking probiotics? Example, you're supposed to drink them every day, because typically a lactobacillus that you introduce survives 5 days and then it's gone. So it doesn't survive longer, so you have to, drink it every day if you want. And then one of the questions that we also like to address is, does it matter which bacterium you take? I mean, is a bacillus different than E. coli, and does that different than this, and so on? So can we see that there's patterns or strategies that bacteria have? So then the question obviously became, how do we actually do it? And I guess that we're a victim of the technology that we had at that point. So we said, okay, let's try to break it down. The most important technology that we will use is transcriptomics, because it's relatively easy to do. So we look at all the genes in the organism and we see if they go up and down, and then we compare it and say, oh, this is interesting compared to what is not there. Then we tried to cut it down in different experiments. So we said, okay, we've worked first with a pure culture for the time being. We work with a complex material. We can discuss about the complexity of this material. For the time being, we said we have to make a compromise because technically you want to put bacteria in and you want to get the RNA out. And in some cases, this is just very, very hard. So it's not very useful to immediately start with the most complex condition. And then we want to have one of those complex systems without a specific carbon source for which this bacterium, of which this bacterium can profit, and the other one with. So if I use a bacterium that likes toluene, then I would have one system, no toluene, and the other one toluene, assuming that this is the specific thing that that bacterium likes. So if it does its job, it will proliferate, and we can actually see it. And then we would like to establish different things. So the idea first being, what can you test? You can put this strain in your complex environment, and then immediately look, does it survive? Because it may die right away, and then that's not a good thing. So we measure the immediate survival, so within an hour. And then we said, okay, we also want to follow growth. So we want to establish as much as possible the same physiology, so the same growth parameters in the sand, in this case, as in a flask. So we hope that we can get the same growth rates. If you don't have the same growth rates, then comparing the transcriptome is already introducing a difference, and then you cannot compare it very well anymore. So we looked at that as well, and then, as I said, most of what we have done so far is measuring by microarray. So let's look at some data. So one of the organisms that we have worked with extensively is this one. It's called Sphingomonas. It's an alpha proteobacterium. You may not have heard of it. It forms, under certain conditions, it forms these rosettes, like also other alpha proteobacteria do. It likes this compound. This is what we call a polycyclic aromatic hydrocarbon. It's called dibenzofurane. It also likes these ones when there's chlorine here and there. And that's why this organism was isolated, because they say, oh, this one can detoxify dioxins, dibenzofuranes, and chlorinated compounds of that. So we looked at what happens if you grow this bacterium in your flask and then put it into soil or sand. That's here and that's shown here. So it matters whether you have actively grown cells or cells in stationary phase. And if you have actively grown cells, you can get back about 80 to 100% directly after introducing it. And that was very important, because if you have too few cells that are alive, then you will only measure death. You will not measure anything useful here. So then we said we have to lay out an experiment where we can actually try to see what is changing in the organism when it comes into such a system. Now, when you do transcriptomes, you always have to figure out a little bit how can you design your experiment so that you can take the comparisons out of it that you want. So we said we are doing 5 different experimental conditions and 2 pre-cultures. So it doesn't really matter how much, but we grew it on 2 different substrates. We put it back in sand, or compared not in sand, but in the same physiological conditions. And here, almost the same thing. Important to realize is that when you're limited by technicalities, like you have to isolate 10 microgram of RNA or so, then you have to start with a reasonable number of cells that you put in the soil, because otherwise you just can't get enough out, you can't get your microarray data, so that's the limitation that we had here. And then they stay 1 hour in there, we isolate the RNA, label it and put it on a chip, and then we compare. Oops, that's the wrong direction, that's not the idea. So I don't know if you ever looked at microarray data. So the problem here, and that's a problem that will become apparent to you, then you will say, oh, this boring guy, what's he talking about, you know? The problem is that you will have a list of 5,500 genes in this case, in this organism. You can display it like this is called a heat map. It doesn't project so nicely as it should here, but it doesn't really matter. You run triplicates, so they should be more or less the same, so you can cluster them. If the triplicates don't look the same, then you can already forget about the comparison. And then you'll want to try and interpret something useful out of this. Now, what I find not very useful is to look at 5,500 genes and say, oh, look, this goes up and that goes down, because it's just too detailed. So one of the problems that you have here is, what kind of language do you actually use to interpret what's going on here? Now, something that's very simple, but that can already be very useful, is just looking briefly at those things and looking briefly at what's called a Venn diagram that just displays how many changes do you get between one or the other condition. Now, the interesting thing is, so if we change to a material that does not carry the carbon source that we want, then something really obvious happens, but it's very interesting to see it. So then your cells will completely interrupt their metabolism. Within an hour, they will start scavenging for nutrients, and they will start their survival program. So for practice, this is a very important point, because very often, you know, here we add the specific carbon source that the cells need. But if you go to a contaminated site, perhaps this material is not immediately, as we call, bioavailable. So then when you bring the cells in, they don't see anything, and they immediately go to their sort of starvation phase. They start to panic and try to get what they want, and perhaps they cannot start sufficiently what you want them to do. The second thing which we saw is that if you grow them on a different carbon substrate than what you want them to degrade, then they have to adjust their metabolism. It sounds logic, but the interesting thing here is that it was proposed that this compound, which is called salicylate, is an intermediate of the other one, dibenzofurane, which apparently, if you grow it on that intermediate, the cells react very differently, and I will show you another diagram of that, and they will have to completely readjust. So also that has some important practical implications, because you may, for reasons of how much this actually costs, you may want to choose for one specific carbon source to grow your cells on. Perhaps sugar is very cheap, and then you go to a polluted site and they have to see something else. But then the cells completely have to readjust their metabolism, and during this time, perhaps they're vulnerable to changes that you don't want. However, if you go from the same substrate to a sand, in this case, with the same substrate, then you see very little changes. And then you see that the cells immediately start to metabolize, they start to respire, they induce a number of biosynthetic pathways, and you can see that they are actively and doing what they are supposed to do. Now, interestingly, and this may be just by chance, if you overlay all the changes between the different conditions, you find that 40 genes actually are conserved at this point. So they are popping up all the time. I don't want to list them here. It could mean that this is sort of a central, you know, like a core set of parameters that the cell needs to react when it encounters something like sand. I'm not going to in detail about it. So then we said, okay, it's nice. So we can see that if you put them in, just putting them in, you can already see quite some changes, and you can also interpret what is going to lead to success or not. Then we said, more importantly is, can they actually grow under certain conditions? We know they can grow in liquid flask, that's shown here, and you get a nice growth curve. But can they actually also grow in sand with this substrate, and that's shown here. So you can see that also in sand you can get growth. I will point out that the scales are not the same here. If you don't add anything, there is also just a tiny, tiny little bit of growth. Now, what is a bit of a nuisance here with these kind of things, and that's very often when you talk about bioremediation. So some of these substrates are unsoluble. So this compound, dibenzofurane, is a powder, it's a crystal. The maximum solubility in water is 10 milligram per liter. So that means that experts will probably recognize that this is not an exponential growth curve, but this is a pseudo-linear growth. And in that case, the physiology of the organism is driven by the diffusion rate of the dissolution of the crystal. So the dissolution rate of the crystal into the water phase, that limits the growth rate of the organism. So when we saw this, we said, okay, what we have to compare here in our transcriptomes afterwards, we measured like the maximum specific growth rates in the exponential phase, but the exponential phase is actually extremely short in both cases. Then you get pseudo-linear growth, and then you have sort of a stationary phase. So we said we want to measure extremely early, and then we want to compare the stationary phase, and we also look at this. Now, there's another thing that is perhaps interesting here. So if you want to look at growth under such conditions, you have to start with a very low population. So previously we started with 10 to the 8 cells per gram. Here we start with something like 10 to the 6. And the reason is, obviously, if you start with 10 to the 8, there you don't see any growth, because the amount of carbon is not sufficient for the cells to multiply. So you cannot see any growth, you have to start really low. But this has the disadvantage that if you want to isolate RNA, you need to, so my student Sylvia, who did this, you know, we worked with microcosms, but that meant to isolate sufficient organisms in RNA. Here you have to work with 160 microcosms, which have to sort of try and extract and pull and then re-pull and so on to get sufficient RNA. So that's a bit of a nuisance. We could also see that the organisms in the sand actually degrade the dibenzofurane, that was shown here, but not to completion. So from a bioremediation perspective, this is not good, but for us it was sufficient to actually look at this. So then we did the same thing, isolating the RNA, triplicates during the microarrays, and then we looked at the thing overall, and that's shown here. So this is a PCA of 5,500 genes under the different conditions. And what it tells us, for example, these are cells that are growing exponentially in sand. So overall, their transcriptome is very, very different from cells growing exponentially in liquid, because these are here. You see? So even without looking at any genes in detail, we can see they're completely different. So this is not the same thing. So we try to work as closely as possible on the same growth rate, just the material is different. But if you look at stationary phase, then the things come closer. So this is stationary phase in liquid, and this is stationary phase in the sand, and then obviously they become closer to each other. Now, then how do you actually interpret these kind of things? So we try to focus on a number of things. But as I said, the problem is really the language. How do you do this? So in one case we said, let's take a look at the metabolic pathway that we expect. So from the gene expression analysis, we figured out a metabolic pathway, and that actually looks more like a network here. So all these boxes, they are different intermediate compounds, and the lines that connect them means this is an intermediate from a particular substrate, goes to a product, goes to another product, another, and so on and so on. So this is until you have acetyl-CoA and succinyl-CoA and so on. And the thickness of this line is a representation of how strongly that gene under that condition was expressed. So this is just comparing exponential growth in liquid and exponential growth in sand, and you don't need to be a biologist to see that there is a lot of difference between the two things. So it means, for us, it meant two things. It meant that, so what we often know about metabolism being something linear may not be true. It looks like a network. And even though the substrate is the same under these two conditions, the cells do something completely different, apparently. So perhaps they decide on the basis of whatever reason that this pathway is more important in the sand than it is in liquid, and this one is totally unimportant in the sand, whereas it is in liquid very important. You know, so there's decisions, and we don't know why they make these decisions. We can only say, okay, this is what we see. So you can also see this a little bit in this heat map where we list all those genes again, but then their expression level. So red is very high and blue is very low comparatively. So you can see this is cells growing exponentially in liquid, and this is exponentially in sand. And you can see these patterns are very different. So the same gene that we know is important for the degradation of this compound is only important in the liquid conditions, but not on sand, whereas the cells, they do utilize the substrate. Okay, so that's kind of curious and tells us, oh, many things are much more complex than we actually think. Now then, how do you interpret these things? So we tried to take advantage of something that's called gene ontology. I don't know if you heard of it. It actually starts from eukaryotic, it started by eukaryotic biologists trying to interpret something in words. You know, like you can have an RNA polymerase, but okay, what's it? So you want to know what's the process that this RNA polymerase is in, that's transcription, and so on. So many of these terms are a way of trying to interpret by words what the genes that you find going up and down actually mean. So these gene ontology work with three different ones: biological processes, molecular functions, and cellular components. So often these end up in the same thing, but it's just taking a different perspective. So if you look at the Go ontology, it's a huge network of things that starts with those three: biological process, cellular components, and then go a cellular process is a metabolism, and a metabolism can be carbon metabolism can be glucose metabolism, and so on and so on and so forth. Okay, so it's a way of trying to interpret it. Now, if you look at these things, what do you say? Okay, let's forget about these numbers. Let's forget about these numbers. What can we interpret? So what you can see is that this list is smaller than this list. That's surprising. So in sand, this is what the cells do specifically in sand. So what they tend to push up. So they need it. What they don't need, apparently, is a longer list of things that they actually try to shut down. Now, for example, what they modify here are parts of their cell wall. We don't know why at this point, but that's what we see, okay? They also change parts of their metabolism. You have to understand that even though there is a word like pentose phosphate shunt, this is always limited by the information that you feed into. Huh? So most of the genome that we know of, 50 to 60% of things are known, 40% of things are unknown. Okay, so the unknown cannot turn up anywhere there, but they may be very important. We just don't know how to describe them at this point. Yeah. So, on the other hand, what goes down are things that are also interesting, like response to stress, motility, the aromatic compound metabolism. That's what I've shown before in these networks. And you'll see that the same thing pop up here. So the process motility goes down, but also the synthesis of the flagellum goes down. So that's kind of the same thing. Okay. So when we looked at it, we said, oh, this is kind of strange. It is as if cells are almost more stressed in liquid than they are in sand. So it seems that they're even more happy when they're in sand with the same compound than in liquid. So perhaps the compound exerts even more stress in what we do under what we think is the most optimal laboratory conditions. Okay. Now I try to interpret it slightly different for you. So if we have different steps, you go from your lab culture, you go to an early transition, you put them in the sand, you look after 1 hour, what do the cells do? Okay, well, what we see is they scavenge nutrients. So they realize that in your flask you may have 1 gram per liter of ammonium chloride or so. That's not the same in the sand. Okay, so they start looking for nutrients. All kinds of transport processes are activated, meaning they want to get stuff in that they perhaps don't see much anymore. They try to maintain what they call homeostasis, whatever that is. You can look in detail more. And there's some evidence for oxidative damage occurring at that point. Now, when you get growth at the gene expression level, this translates into, yes, you see genes popping up involved in energy generation, ATP synthesis. You see lots of things being biosynthesized, which you would expect when cells are actively growing. But also you see things that are not immediately easy to interpret. So this organism has 80 proteins that are called TON-B receptors. Very little is known about these TON-B receptors, except that they're sort of scavenging importers for a variety of things. So we see a lot of them being higher expressed under those conditions. Then we also see a lot of factors that may be implicated in adhesion, at least that's what we think, like pilins, components of a type 4 secretion pathway, so also things on the surface that we see. And then in the later phase, so which we call the survival phase, we see, again, a lot of things that we can interpret like, okay, there's a diminished expression of energy generation, probably because they run out of their immediate food source. There's diminished biosynthesis, so they stop growing. That's what we also see at the micro level. We see increased stress responses, nutrient scavenging, turnover of proteins. We see increased transport and synthesis of polysaccharides, perhaps to protect the cell and to prevent their drying out. And also then we see like production of osmoprotectants. So I find this kind of interpretation useful because it's like saying in words what these microarray data are telling you. So in this case, we can say, yes, the organisms can survive such a transition, and this is how they will react. Now the question then is, obviously, so how important is this, what we see? Okay, so how can you look at this? How can you actually understand something like how important that this is? So we try to do a second type of study, which is try to interpret which gene functions are possibly important for the survival or the fitness of the strain, then compare it back to the microarray data to see what we can make of it. So here we're using a technique which you may not have heard of at all. It's called transposon sequencing. You may have heard of transposons. So geneticists can use transposons to actually damage genes. So you induce them in a cell and you will damage genes at random. So what you hope to get is a library where in every cell there's at least one thing damaged, so that when you have the library sufficiently large, that the library as a whole has everything damaged once. Okay? So for example, we work with a library of 20,000 mutants, by which statistically we can say, okay, on average, every function should be hit 3 times. Because there's 5,000 functions, 3 to 4 times, so we may have seen everything. Then the idea is that you can actually grow this library of mutants under different conditions, condition A, condition B, and what the technique allows you is to look at the whole library and quantify exactly how many of each individual mutant do you have. Now, if condition A or condition B has a different fitness effect on certain mutants, those will disappear from your meta population, and you will see a diminished occurrence. So you will compare those things quantitatively, you can plot them in a graph, and then you say, oh, look, this particular function is disappeared compared to my starting library. So this was an essential function at this particular condition. Yeah. Now, it looks a little different if you do that for 6,000 genes, but I'll guide you through. So we did another experiment again with this Sphingomonas vitihi. We created 2 libraries, each of 20,000 clones. And then we said, okay, we do a simple lab experiment as comparison. So 50 generations, transfer one flask after the other. And that would create certainly differences in the transposon library, because anything that the cells cannot use or that leads to a fitness loss on those conditions, these mutants, you will not find them anywhere. And then we also said we do the same thing in sand again with an aromatic compound. Here we thought of it, but we couldn't really come up with something useful to actually do repeated transfers. So we said, okay, how do you do, from a flask you can grow one batch culture, you have 10 generations, you can take a sample and put it in next liquid, but from soil that's kind of difficult. We couldn't really find a good way of doing this, so we said just grow them in sand, and we do 2 conditions: 4 days incubation, in which case you have about 7 generations, and then 10 days in which you have not really much more, because you can see that the cells are actually declining. But this, interestingly, was already sufficient to get lots and lots and lots of mutants. So then in reality it looks like something like this. So this is only one part. This is only showing 300 genes, but you have the starting conditions with the abundances of the different individual mutants. You compare them to one condition, you compare them to the other condition, you normalize their numbers, and then you divide, and you can say by using certain cutoffs. So these genes are not impacted, but these ones are overrepresented under this condition, and this was underrepresented, and then certain genes you will find no mutants ever because they're essential, right? And then you can try to actually interpret what that means. Now, how do you interpret again what that means? So again, it's a problem of language, right? So we try to use a bit the same kind of tools. So this is just looking at numbers. And you can see here from these comparisons of different conditions, so in liquid flasks and then in sand, you can see the numbers of specific genes that you find cause a fitness effect under those conditions. For me, it was enormous, like 600, that's 10%, that's almost 10% of the genome is specifically affecting the fitness only in sand, not in liquid, nothing, only in sand. So it's a lot. It tells us there must be lots of functions that the cells really need for those conditions specifically. And depending on how you look at it, so we looked at tenfold underrepresented or completely absent. This number is actually, when you add it up, is even larger. But you can see absent genes, so there's an enormous amount of mutants that disappear completely even after 7 generations of growth in soil. Okay, so not 500 generations, just 7. That's I think that's enormous. Now then you can actually look at this in a particular way, because the question is, so is this significant or not? So what we try to do is then look at those numbers and the genes and classify them in particular categories. So one of the categories that's also used is called the COG, for cluster of orthologous groups. It's again an effort to say, okay, I have here a gene for an RNA polymerase. What category is it? Transcription. Okay, so we put it in the box K for transcription. We have another thing that's important for signaling, so we put it in the box T, and so on. And then you can add all up and normalize for all of the categories that you have, and then you can compare it, so you can see all the genes in the genome. If you put them in those boxes, you get a certain representation. Okay, like okay, 8% of the genes in the genome fall in this particular category K. Now you can do the same thing with the genes that you find cause a fitness effect under those conditions, and compare that distribution with the distribution of the genome, and then you can say, like, oh, look here, in this case, we find an overrepresentation of genes in this category that are important for fitness under those conditions. Yeah? So that helps you to see, and you can see that there's a lot of categories where you find overrepresentation of genes that cause fitness loss under those conditions. Right. Now you can look again at a list, which I won't do, although there's certain things that you will not see in COG categories, but you only see them if you look at Go terms, which I find a little bit clearer. But I want to highlight a few things that you cannot capture very well in these categories or so. And these are things like, okay, we found that there's a very specific protein that nobody knows much about, which is extremely important for these cells. It's called a surface antigen. And this is very highly expressed under sand, and it's also absolutely essential. If you take it away, cells cannot. So this is this cell shape remodeling that we think is also extremely important. And then there's things that we didn't thought at all would be important, like arsenic detoxification. It's absolutely essential. If you go to a soil here, if it doesn't have this fitness loss, you find extremely high expression. And then we also see things like, which is also intriguing, flagellar clusters. So this relates to what we have seen before with this motility. So the cells generally tend to reduce their motility, but on the other hand, the flagella is essential. So it's kind of bizarre. So the flagella is essential, but they reduce motility in the soil. But if you don't have flagella, then the fitness is extremely reduced. And interestingly, so this organism also carries two big plasmid DNAs, and also several functions on the plasmid DNAs are essential for the survival in the soil. Okay, so I can see that you are sort of already getting saturated by all these terms and things and so on. So what are we currently trying to do? So I think, and that's something that I am hoping that we can discuss, is so what kind of language can you use to describe these things? Or do you have to put it in a model, or do you have to put it in a predictive thing, or so on? Now, the next step for us are actually two things that we are working on. So one is sort of a meta-comparative level, because one strain, this could be anything. How can we generalize from these things? So we said, okay, can we actually do an experiment? It's a lot of work, and we're still working on it, where we use 3 completely different organisms. So this is an alpha proteobacterium, this is a gram-positive bacterium, this is a gamma proteobacterium. So very different bacteria that degrade compounds that we are interested in, that occur very frequently: benzene, toluene, ethylbenzene, chlorophenol, a pesticide, and dibenzofuran, which you find a lot. So all of these bacteria could be interesting. So we said, how can we ever compare those? So we're trying to use sort of the same standardized stress conditions. We still want to do this experiment in soil, but it's a lot of work. So we said we can do something more simple at this point, and that is we try to recreate what we think is an important variable in sand or soil, and that is drought stress. Okay, because very frequently the water content of the sand is not sufficient, and this is something that cells react to immediately. So you can mimic that in liquid culture by 2 types of stresses. One you induce by adding a salt, which is called solid stress. One you can induce by adding a swelling agent like polyethylene glycol. That's called matrix stress. So there are different kind of stresses, but the resulting effect is that the cells will think that they have less water availability and they will react to it. So we were hoping that this actually mimics an important point of what we see in soil. It doesn't, but that's something else. Okay, so then we measure like what's the survival of the strain under those conditions when we decrease the water potential to a different level. And then we choose a level like here where we see that the cells don't die, because if cells die, you cannot measure anything anymore. But where they are like 20% impacted by the treatment, okay? Because then they're still alive, but they have to do something because we see that they react. And then we do the same thing as before. We expose them to them, and then we isolate the RNA, and then we look at what they are doing. So we try to do that for 3 different strains now. So they all, at some point they are affected by drought stress. But this organism, this Archaeobacter, is very peculiar, because if you look at, just the numbers, so Pseudomonas and Sphingomonas, they're very sensitive. So -1 megapascal water potential loss, they are sort of struggling to survive, whereas Archaeobacter can go down to -5 megapascal, and then it's still sort of surviving. So this one already seems to have a different behavior. And that's represented if you just look at the numbers of genes that we see going up and down. So Sphingomonas, you see hundreds of genes under those conditions, but this Archaeobacter, only dozens of genes. So somehow it doesn't even realize that there is something like drought stress. That's very peculiar. And this one is sort of in the middle. Now then you can try to see what are the things. But then comes again the question, how do we compare it? I don't want to bother you with, again, lists of things, but we have to find a way to actually compare it. So we said one of the things that we can do is, these are networks, actually. So this is a network of gene ontology, again, whereas this is the highest node, and there's lines, they don't project very well, so these lines connect from, this is the highest node biological process, and then it goes to a metabolic process, and so on and so forth, so it goes all the way down. And then we plotted on top of this the major changes that we see in each of the organisms. So in 3 colors. So the blue one is the Archaebacter, the pinkish one is Pseudomonas, and the white one, bad choice here. The white one is Pseudomonas vironi. So you can see in some cases, like here, all 3 react in sort of a similar way. But in many cases, like this one here, you see only pink spots. So that's very typical for one strain only and not for the other. Now, what are the things that you can see that are always the same? It's motility, flagellosynthesis, and chemotaxis. So this we find always under those conditions. So no matter which of the 3 strains we take, they will reduce under drought stress. They will reduce the expression of the things that have to do with motility. And this is interesting because it has been shown that motility and flagella are really, really very important for the fitness and the long-term survival in soil. And then other things that we see, one other thing that I want to highlight is the effect of stress and DNA repair. That's here for this Pseudomonas vironi and here for Sphingomonas and Pseudomonas vironi, but not for Archaebacter. So there it's not stressed at all. Okay? So this is the kind of thing that we try to use to actually interpret what's going on. Good. So some final words, perhaps. I have some more stuff, but I prefer if we could actually discuss something. So when I was talking to Victor and Stan on Friday, they talked about understanding. And Stan, he says, well, what is understanding? I never thought of it. What is understanding, right? Is it a description of things, or is it a mathematical formula? Is it a prediction of things? So what do we actually understand? So I thought the things that very general things that came to my mind is that, okay, if you put a bacterium back into a complex environment, it realizes it. You know, it's not the same thing as a liquid. So many microbiologists, they are focused on their reactors and culture flasks and so on. At the level of what we see for the programs that the cells are running, this is not the same thing, even though they can grow to with the same rate. You know? So I think this is an important message that, okay, if we want to do these kind of studies, we have to be aware that, you know, a liquid culture is just one very, very peculiar condition, and it's not at all reflective of things that are going around in the environment. So in terms of what we see, we find both very trivial reactions, like, okay, when you give the cells nothing to eat, okay, they stop. You see it immediately. You know, it occurs in half an hour. So that gives you a message. And then when they start growing, we see all kinds of biosynthesis, energy generation, as you would expect, but you see it happening. And then there's specific functions which we also see, which we don't know, like this specific adhesin or so. What's it good for? Nobody knows about this adhesin. So that could be interesting just to try and understand what's doing. So if we look at 3 different genera, the responses have very little in common to the same kind of stress condition. That also perhaps limits our ability to actually find conditions where we can have a successful remediation, or perhaps it tells us this organism we should use, others we should not. And then I find it extremely difficult to find a language. I mean, I can see puzzling faces here already. You know, a language. How do you look at the gene expression data, proteome data? If we have all these data points, but in the end of the day, what you want to say is, okay, the organism is happy, or it's not happy, or it's dying, or it needs this, or it needs that. So you have to find the language to actually approach it to this level. And then, yeah, what is understanding actually? I mean, we have a lot of descriptive data. So if you try to publish this, then people will say, descriptive. Well, is that bad? I don't know if that's bad. Is it predictive? Well, some trivial things are perhaps predictive. Should it be predictive? I don't know. If you wanted to predict it, what are you looking at? Is a math formula, is that a proper understanding? Or is that just also a description which you can stick in the computer and you get an outcome, but you don't really understand what that means, right? So I think with that, I probably stop. I have some more things, more slides just at the single cell level, but that doesn't really matter so much. And then I think it's preferable if we should discuss about this. Okay, thanks a lot for your attention. I'll be happy to discuss. Right. Yes.
Did you try to look at the cell morphology and the cell condition and the normal? Because it looks like you have geos in flagellum and stuff like that. Do they form any biofilms and something with sand or?
Well, you know, it's an interesting. So there's one anecdote which I would like to share with you. I don't have a picture here, but it's a very strange thing. So Archaeobacter is an amazing organism. When we did these tests with the, for example, changing the water potential, this organism can change its morphology instantly. Okay, you could look under the microscope, you can put a salt droplet on the side. When it's diffusing into where the cells are, they shrink. And it doesn't affect them at all. It doesn't change their expression, gene expression. This is totally amazing. I don't know how to interpret this, but that's just an anecdote. In terms of the soil, you know, it's very difficult just because of the technicalities. How do you do it? If, I mean, I can show you pictures of how cells look like in soil, but they look like, they look like cells to me. They look like what we expect in liquid culture, mixtures of things. But this is what we need to do here is we have the soils, we have to extract the cells out of it and then put it on a microscope slide. So I don't know any method where you could actually look at how the cells look like in such a matrix. It's impossible. It's not transparent. I've never come up with a good idea on how actually to look at the cell. Do they change their morphology or not? We have seen things that's also very strain specific. So like I said, under certain conditions, this strain will form these rosetta. And I don't know why, but that may, so there's people that think that they stick at the poles for some reason. But biofilms at this level of community sizes are, I think, relatively unlikely. So it's 10 to the 8 per gram of soil. I don't think you have biofilms at this point, but I cannot, you know. Yeah, it's like what kind of experimental tools would you do?
Well, I don't know.
Yeah, no, but we have thought about the same type of thing. So how can we get a handle to see what the cells are actually doing in something like a soil? And I just can't come up with any good experimental tools. You can make a very thin soil layer on a microscope glass and something like this, but you always have focusing problems, so you cannot get really where the cells are. I mean, here we're using gene reporters to visualize them, but also there, you know, if they're not in your focal plane, you cannot quantify, visualize them anymore. So it's really very hard. It's an interesting question, but on an agar surface, yes, but in the soil, extremely hard. I don't know how to do it. Yeah.
For the degradation of some of these compounds like TBF, do all the strains use the same pathway of TBF degradation, or are there different pathways that they can choose depending on the conditions?
Yeah, you mean like sort of job sharing among a population? Yeah. So we try to do something about this. I think many people are interested in this. The problem. So that was also one of the reasons why we look at reporter genes, which you can stick to specific promoters and so on. The problem is what's the natural variability, so the variation in this expression that you have on top of something else. And then with many of these organisms, the problem comes they are not model organisms, so you cannot do all the genetic tricks that you want. That has limited us a little bit in what you can do. From these network things, we have the feeling that it could very well be that actually it seems sort of logic, but it's also here, it's like, where was it? Sorry. Yeah, here. Oh, here. Yeah. So when you just think about it, how can one cell operate with different parallel pathways? I find that difficult to believe. Perhaps it's possible, but we'll have to ask biochemists if to know if this is possible, that you can operate multiple pathways at the same time. The alternative would be, yes, some cells are using this, the other ones are using that, and so on. So a community shares this, but the only way of measuring this, so this suggests that cells are doing different things, and perhaps in soil they become more differentiated over multiple pathways, perhaps because they want to profit of other carbon sources that are laying around as well. But how do you measure it? You know, it's an interesting question. I think it bothered me for a long time. This Victor can perhaps comment on this idea of having linear pathways or branched things where cells can sort of profit from having multiple possibilities, and perhaps they profit from this in a population by having certain cells doing this and others doing that. Then the question becomes, how do they know what to choose? You know. So, but,
But it would be very interesting to go in further in depth into his with microarray experiments, because then you can really picture.
The problem with microarrays is that you need a certain quantity of cells. So if that quantity of cells is, if you cannot have it, then you cannot do it. So there's also something that's a single cell RNA sequencing that exists for yeast, but it doesn't. I haven't seen any real reports for bacteria that this should be possible, you know, because you have to, and then the problem is you have to amplify the cDNAs and so on, so you get a lot of rubbish. I think that the reporter systems are the best for now, but you can only look at 2 or 3 parameters at once, and you have to be able to actually manipulate the strain sufficiently well that there's no bias coming in from anything there. And this is a horrible organism in that respect, unfortunately. Although, I mean, it's being a wild-type organism is very fascinating as well. Yes.
These diagrams that you have here, I mean, just looking at it naively, it looks as if you've just got less going on the right than the left.
Yes.
But that can't be true because you've got the same growth rate, right?
Yes, but it doesn't need to be reflected in, you know, this is difference in gene expression. So the problem with gene expression is that it's not one-to-one related in protein levels.
It's something else.
No, this is not flux. This is just plotting because we don't know. I mean, for measuring the flux, you would need like C13 labeled material. So this is only measuring this is plotting the change in gene expression of this particular function
So this enzyme,
on that pathway. Yeah, the gene for that enzyme on top of it.
sometimes you seem to have a very dark line and then a very small line. So it seems like it upregulates an upstream enzyme and then it
Exactly,
doesn't bother to
yeah.
upregulate the
Yeah,
acid
but that
side.
comes back to we all have in our minds the toll pathway that Victor knows all about, that has 2 regulators and everything works. But in this case, these genes are scattered over, well, we calculated here once, I think it's 55 different transcription clusters.
Mhm.
So the regulation is so different that may well be you get here very thick and then it's thin, because that's just a gene that's in a different part of the chromosome. It's regulated differently.
Going back to the acrobatic story. So what you are saying is that you don't see much changes in gene expression, but you see massive mechanical changes.
Yeah, yeah.
So so these genes, the few genes that you see, do you see anything interesting in them, like tetra halos or anything like that, or is something non-trivial to interpret?
For now, it's a little bit non-trivial to interpret. I mean, we have focused on certain things that we would like to see, such as osmotic protectant or so. Yeah, we see that kind of thing. And in other cases, I think we're just biased by what we want to see and not necessarily exactly what is there. So for me, this is always a bit the danger of the things. No, you can interpret what you know already, but the unknown parts are much more.
But is acrobatic resistant to desiccation? I mean, you can leave them in complete dryness and it will still survive? Or
Pretty much, yes. This is what they did in Sweden, no, with this putting it on this vermiculite material. Yeah, so it's pretty resistant to doing just that. Yes. Or okay.
I was just interested in for your sand versus liquid culture experiments. For your sand, did you add other nutrients like you would have done for your liquid culture? Were they comparable in that way, or were they just natural sand from a certain place?
No, we try to have the same. The difficulty comes from the water content of soil, right? Because in a liquid, you can have the same concentration as what you assume to the liquid that's in sand, but the total amount will be different, right? If you would have the total amount, then the concentration is different in the soil, in which case you get the over salinity. So we start with in this case we start with the same concentration of mineral components, but that may mean that at some point in the sand they run out of their mineral nutrients. We didn't find any good compromise of avoiding that actually the osmolarity in the sand is higher compared to liquid, because that would have immediate consequences, as a matter of fact. Yeah, but it's that's a bit, again, how do you do this? Because you know that in your microcosms the water content is not so high, and that means you don't have a lot of liquid to start from. So we focus on having the same concentrations and not the same amounts as compared to the liquid culture.
Hi,
Yes.
yeah. About these two graphs, so are these cells have the same growth rate, different growth rate?
They have the same growth
Same
rate.
growth.
Yeah.
How about the rates of DBF degradation? Are they same or different?
It's difficult for us because the problem is that in liquid culture, so oops, what's this one? So what we try to do here is, because the dibenzofuran is insoluble, so we try here to add the same quantity of the culture. But if you have, the optical densities here, for example, are so much higher that we add a lot more, so we add saturation amounts of dibenzofuran. So we cannot really calculate a rate of dibenzofuran degradation from this particular experiment, whereas here we try to do it quantitatively to at least demonstrate that yes, the cells do something in the sand, they're not just growing on something else which the sand has. So I cannot really answer your question if the degradation rate of dibenzofuran is the same as in liquid culture. Because for that we would have to do an experiment with a limiting amount of dibenzofuran.
As to the width of each individual line, are you comparing on a per cell basis or
The width of what? Sorry.
Can you go back to the graph?
This
the,
one?
The diagram, the box diagram, the graph. The first one that you showed. Yep.
This one?
Another one.
The network.
The network.
The network.
Okay.
The network.
Yeah. So,
Here, the width of the darkness of the line.
yeah.
Are they have the same in terms of same quantities per cell base or the per culture? What's the what's the unit of these lines?
The unit of these lines is a relative unit, because this network program, I mean, you can actually, it depends on how you want to display it, whether you display on a linear scale the width of a line compared to the gene expression level that you see, which we didn't do here. So this is not on a linear scale, these lines, okay? Because otherwise you cannot display all the lines in the same level. So this is an exponential, basically line thickness increase compared to the gene expression. But then everything is standardized through the amount of RNA that we isolate. And not necessarily the amount of RNA comes from the same amount of organisms, because from the sand this is very hard to determine, because you may lose you may lose cells. So that's why we only standardize to the amount of RNA that we have.
So if I understand this correctly, that means the same width or same darkness of the line in A doesn't mean the same darkness in B.
Yes, it does. But the same darkness of this line doesn't mean the same, that doesn't mean the same expression as here. Because the genes by themselves are expressed to very different levels on the chromosome. Okay, so this comparison is valid, not necessarily this gene is expressed at the same level as that one, because that can be scaled according to the total gene expression of that gene. Okay.
Very interesting graph.
Yeah, but it's again, you have to make compromises between what those programs are doing and what you want to show in this particular case.
Just a comment about these two. I find it's very mysterious to try to understand why the difference. One possible explanation would be the flux, how the cell regulates the flux under two different conditions. But again, I'm not sure. It's very important.
I cannot help it. I mean, you see the same thing on this one already, that the gene expression patterns are completely different. But then again, it doesn't mean that if a gene X is expressed to a level A, that doesn't mean that it doesn't produce sufficient protein to actually
Anything about try to analyze the metabolites, their relative abundance.
Yeah, no, we don't have the capacity. Unfortunately, we already had problems in getting the dibenzofuran analysis, because this required going to a specified lab that can handle C13 labeled standards and so on to get everything. Would be nice, but yeah, I'm not really sure. That would be very expensive right now. Yeah.
Did you try to do experiment with the varying oxygen with those just in these, for these bacteria? Because, or in general, are these two cultures are two anaerobic cultures, or are they respiring the same way?
Well, actually, interestingly, so where is this?
Looks like that just the genes are.
Well, initially we selected the conditions that we had the best survival, so that's here. The water content that has the best survival. So we said for the subsequent experiments we have to stay under the same conditions, otherwise we run into problems with the experiment. But interestingly, if you compare it to liquid cultures, we see that the cells turn off the oxygen respiration, because in sand it appears they have much more oxygen than in liquid, which is sort of counterintuitive. You would think it becomes anaerobic, but I think that because the sand is not saturated, you have sufficient open pores that the oxygen diffusion rate is actually much higher. And that's why the cells seem to be doing much better in the soil in terms of the oxygen availability than in liquid culture.
It would be just nice to compare if you, let's say, grow in liquid culture but with a different aeration.
Yeah.
See,
Well,
maybe this can explain the phenomenon.
Sure, but 1 microarray is still $1,000. So, so there's a limited thing that you can do. But yeah, there's lots of other questions that would be interesting to answer.
Those metabolites or genes were connected to those metabolites. It's fumarate, succinate. So in that
Diagram.
Yeah.
So the thing that finally comes out.
Yeah. So
it's like acyl-T-CoA, succinyl-CoA, pyruvate, those kind of things.
So they're close to TCA cycle, or
that's the idea with most of the strategies that these organisms have. So they will channel everything down to this TCA cycle if they have one. So these mostly have one, but you always have to be a little bit careful of the annotation, of the genome annotation. So there may be things that are called enzyme X on the base of that homology, and in the end they're doing something different. So. Good.
Actually 3 o'clock already, so
Yeah.
maybe we should,
Thanks a lot.